Hi everyone, in this video we are going to continue with our previous lesson that is the gastrointestinal system and related things. So in this video we are going to learn about the salivary glands and secretions associated with that. So before moving on to the lecture, let's start by watching which are the learning objectives. List the general principles of alimentary tract secretion. List the different salivary glands. Describe the composition of saliva. Describe the mechanism of secretion of saliva. List the functions of saliva. Outline the regulatory processes involved in the secretion of saliva. So these are the learning objectives. Now let's learn some of the major terms related with this topic, like a small review before going on to that topic. Like we will be learning about those terms for better understanding. Like we are going to look at the glossary. Like acini, the plural of acinus, which refers to a small sac-like dilations composing a compound gland. Enzymes, several complex proteins that are produced by cells and act as catalysts in specific biochemical reactions. Next, mucus, a thick secretion composed of mainly of water, electrolytes, and a mixture of several glycoproteins, which themselves are composed of a large polysaccharides bound with much smaller quantities of protein. Then, ptyalin, a form of amylase or enzyme found in the saliva of humans and some other animals. This enzyme helps catalyze the hydrolysis of starch into maltose and dextrin. ptyalin, ptyalin. Then tactile stimulation, stimulation of cells or tissues by the sense of touch. Now let's learn about the major topic, salivary glands and secretion. Throughout the gastrointestinal tract, secretory glands subserve two primary functions. Digestive enzymes are secreted in most areas of the alimentary tract, from the mouth to the distal end of the ileum. Mucus glands from the mouth to the anus provide mucus for lubrication and protection of all parts of the alimentary tract. Most digestive secretions are formed only in response to the presence of food in the alimentary tract, and the quantity secreted in each segment of the tract is usually the precise amount needed for proper digestion. Furthermore, in some portions of the gastrointestinal tract, even the types of enzymes and other constituents of the secretions are varied in accordance with the types of food present. So, general principles of alimentary tract secretion. Types of alimentary tract glands. Several types of glands provide the different types of alimentary tract secretions and basic mechanisms of stimulations of the alimentary tract glands. Contact of food with the epithelium stimulates secretion, functioning of enteric nervous stimuli. The presence of food in a particular segment of the gastrointestinal tract usually stimulates the glands of that region and adjacent regions to separate and secrete moderate to large quantities of juice. In addition, local epithelial stimulation also activates the enteric nerve system of the gut wall. The types of stimuli that activate this system are as follows, like tactile stimulation, chemical irritation, distension of the gut wall. The resulting nerve reflexes stimulate both the mucous cells in the gut epithelial surface and the deep glands in the gut wall to increase their secretion. Next, autonomic stimulation of secretion. Parasympathetic stimulation increase the alimentary tract glandular secretion rate. Stimulation of the parasympathetic nerves to the alimentary tract almost invariably increases the rates of alimentary glandular secretion. This increased secretion rate is especially true to the glands such as the salivary glands, esophageal glands, gastric glands, pancreas, and adrenal glands and the duodenum. It is also true of some glands in the distal portion of the large intestine, which are innervated by pelvic parasympathetic nerves. Sympathetic stimulation has a dual effect on the alimentary tract glandular secretion rate. Sympathetic stimulation can have a dual effect on secretion. Sympathetic stimulation alone usually slightly increases secretion. If parasympathetic or hormonal stimulation is already causing secretion by the glands, Superimposed sympathetic stimulation usually reduces the secretion, sometimes significantly so, mainly because of vasoconstrictive reduction of the blood supply to the glands. Next, regulation of glandular secretion by hormones. In the stomach and intestine, several different gastrointestinal hormones help regulate the volume and character of the secretions. These hormones are chemically polypeptide or polypeptide derivatives and are liberated from the gastrointestinal mucosa in response to the presence of food in the lumen of the gut. The hormones are then absorbed into the blood and carried to the glands where they stimulate secretion. This type of stimulation is particularly valuable to increase the output of gastric juice and pancreatic juice when food enters the stomach or duodenum. Now, basic mechanism of secretion by glandular cells, secretion of organic substances. Although all the basic mechanisms by which glandular cells function are not known, experimental evidence points to the following principles of secretion. The nutrient material needed for formation of the secretion must first diffuse or be actively transported by the blood and the capillaries into the base of the glandular cell. Many mitochondria located inside the glandular cell near its base use oxidative energy to form adenosine triphosphate. Energy from the ATP along with the appropriate substrates provided by the nutrients is then used to synthesize the organic secretory substance. The synthesis occurs almost entirely in the endoplasmic reticulum and Golgi complex of the glandular cell. Ribosomes adherent to the reticulum are specifically responsible for formation of the proteins that are secreted. The secretory materials are transported through the tubules of the endoplasmic reticulum, passing in about 20 minutes all the way to the vesicles of the Golgi complex. In the Golgi complex, the materials are modified, added to, concentrated, and discharged into the cytoplasm in the form of secretory vesicles, which are stored in the apical ends of a secretory cell. These vesicles remain stored until nervous or hormonal control signals cause the cells to extrude the vesicular contents to the cell surface. This action probably occurs in the following way. The hormone binds to its receptor and through one of several possible cell signaling mechanisms increase the cell membrane permeability to calcium ions. Calcium enters the cells and causes many of the vesicles to fuse with the apical cell membrane. The apical cell membrane then breaks open, thus emptying the vesicles to the exterior. This process is called exocytosis. Water and electrolyte secretion. The second necessity for glandular secretion is secretion of sufficient water and electrolytes to go along with the organic substance. Secretion by the salivary glands provides an example of how nerve stimulation causes water and salts to pass through the glandular cells in great profusion, washing the organic substance through the secretory border of the cells. At the same time, it is believed that hormones acting on the cell membrane of some glandular cells also cause secretory effects similar to those caused by nerve stimulation. And we just look at the typical function of a glandular cell for formation and secretion of enzymes and other secretory substances. We can see capillary is there, now fiber, endoplasmic reticulum, Golgi apparatus, and secretion will be happening. So that's it. Anatomical types of glands in the alimentary tract and their functions. Like anatomical types of glands, single cell mucous glands called simply mucous cells or sometimes couplet cells located on the surface epithelium in most parts of the gastrointestinal tract. Its function? They function mainly in response to local irritation of the epithelium. They extrude mucus directly onto the epithelial surface to act as a lubricant and also protects the surfaces from excoriation and digestion. Next, invaginations of the epithelium into the submucosa called pits located on many surface areas of the gastrointestinal tract. In the small intestine, the pits called crypts of Liberkin are deep and contain specialized secretory cells. Deep tubular glands in the stomach and upper duodenum. A typical tubular gland can be seen, which shows an acid and pepsinogen secreting gland of the stomach, like oxyntic gland. Complex glands such as the salivary glands, pancreas, and liver. These glands lie outside the walls of the alimentary tract and in this differ from all other alimentary glands. They contain millions of acini lined with secreting glandular cells. These acini feed into a system of ducts that finally empty into the alimentary tract itself. Lubricating and protective properties of mucus. Mucus is a thick secretion composed mainly of water, electrolytes, and a mixture of several glycoproteins that are composed of large polysaccharides bound with much smaller quantities of protein. Mucus is slightly different in different parts of the gastrointestinal tract, but in all locations it has several important characteristics that make it both an excellent lubricant and a protectant for the wall of the gut. Properties of mucus in the gastrointestinal tract. Mucus has adherent qualities that make it adhere tightly to the food or other particles and to spread as a thin film over the surfaces. It has sufficient body that it coats the wall of the gut and prevents actual contact of most food particles with the mucosa. Mucus has a low resistance for slippage, so the particles can slide along the epithelium with great ease. Mucus causes fecal particles to adhere to one another to form the feces. Mucus is strongly resistant to digestion by the gastrointestinal enzymes. The glycoproteins of mucus have amorphoteric properties, which means that they are capable of buffering small amounts of either acids or alkalis. Also, mucus often contains moderate quantities of bicarbonate ions, which specifically neutralize acids. This mucus has the ability to allow easy slippage of food along the gastrointestinal tract and to prevent excoriative or chemical damage to the epithelium. Next, secretion of saliva. Salivary glands and secretions. The principal glands of salivation are the parotid, submandibular, and the sublingual glands. In addition, there are many tiny buccal, labial, and glossopalatine glands. Daily secretion of saliva normally ranges between 800 to 1500 ml, and as shown by the average value of 1000 ml in the table. And saliva contains two major types of protein secretion: serous secretion that contains ptyalin and alpha amylase, which is an enzyme for digesting starches, ptyalin. Then mucus secretion that contains mucin for lubricating and for surface protective purposes. The parotid glands secrete almost entirely the serous type of secretion. Serous in the sense ptyalin enzyme, which is very essential for the digestion of starches, whereas the submandibular and sublingual glands secrete both serous secretion and mucus. The buccal, labial, and glossopalatine glands secrete only mucus. Saliva has a pH between 6 and 7, which is a favorable range for the digestive action of ptyalin. Composition of saliva. Saliva contains especially large quantities of potassium and bicarbonate ions. Conversely, the concentration of both sodium and chloride ions are several times less in saliva than in plasma. So when we just look at the composition of saliva, we can see water and solutes. Solutes in the sense there will be inorganic components and organic components. When we just learn more about the inorganic components, you can see there is sodium, chloride, potassium, bicarbonate, calcium, phosphate, and thiocyanate. These are the inorganic components. Next, organic components. Alpha amylase, which is very important for the digestion of starch, that is nothing else but the chylin enzyme. Then mucin, proline-rich proteins, secretory IgA, lipases, lysozymes, peroxidases, histamines, scatherin, kallikrein, and lactoferrin. One can understand the special concentrations of ions in the saliva from the following description of the mechanism of secretion of saliva. So, mechanism of secretion of saliva. Secretions by the submandibular gland, a typical compound gland that contains acini and salivary ducts. I'm not sure about the pronunciation of acini, but pardon me if I just turned out to be wrong, and check the pronunciation if you need better clarification. I'm sorry. So, salivary secretion is a two-stage operation. The first stage involves the acini, and the second stage involves the salivary ducts. The acini secrete a primary secretion that contains chylin and or mucin in a solution of ions with concentrations not greatly different from those of typical extracellular fluid. As the primary secretion flows through the ducts, two major active transport processes take place that markedly modify the ionic composition of the fluid in the saliva. First, sodium ions are actively reabsorbed from all the salivary ducts, and potassium ions are actively secreted in exchange for the sodium. Therefore, the sodium ion concentration of the saliva become greatly reduced, whereas the potassium ion concentration become increased. However, there is an excess of sodium reabsorption compared with potassium secretion, which creates electrical negativity of about minus 70 megavolt in the salivary ducts. This negativity in turn causes chloride ions to be reabsorbed passively. Therefore, the chloride ion concentrations of the salivary fluid falls to a very low level, matching the ductal decrease in sodium ion concentration. Second, bicarbonate ions are secreted by the ductal epithelium into the lumen of the duct. The secretion is at least partly caused by the passive exchange of bicarbonate for chloride ions, but it may also result partly from an active secretory process. The net result of the transport process is that under resting conditions, the concentrations of sodium and chloride ions in the saliva are only about 15 m equivalent per liter, each about 1/7 of 1/10 their concentrations in plasma. Conversely, the concentration of potassium ions is about 30 MEq by L, 7 times as great as in plasma. During maximum salivation, the salivary ionic concentrations change considerably because the rate of formation of primary secretion by the acini can increase as much as 20-fold. This acini secretion then flows through the ducts so rapidly that the ductal reconditioning of the secretion is considerably reduced. Therefore, when copious quantities of saliva are being secreted, the sodium chloride concentration is about from half or 1/3 of the plasma, and the potassium concentration tries to only 4 times that of plasma. Functions of saliva. Under basal awake conditions, about 0.5 ml of saliva, almost entirely of the mucus type, is secreted each minute, but little secretion occurs during sleep. So, function of saliva for oral hygiene. The secretion plays an extremely important role for maintaining healthy oral tissues. The mouth is loaded with pathogenic bacteria that can easily destroy tissues and cause dental caries. Saliva helps prevent the deteriorative pressure in several ways. The flow of saliva helps wash away pathogenic bacteria as well as food particles that provide their metabolic support. Saliva contains several factors that destroy bacteria. One of these is thiocyanate ions, and another is several proteolytic enzymes. Most important, lysozyme, that is an attack the bacteria, aid the thiocyanate ions in entering the bacteria, where these ions in turn become bactericidal and digest food particles, thus helping further to remove the bacterial metabolic support. Then saliva often contains specific significant amounts of antibodies that can destroy oral bacteria, including some that cause dental caries. In the absence of salivation, oral tissues often become ulcerated and otherwise infected, and caries of the teeth can become rampant. So when we just try to look at the salivary glands and surrounding structures, we can learn that there are branches of facial nerve, then transverse facial artery along with the accessory parotid gland, then parotid gland, then later sublingual fold with opening of sublingual ducts, next sublingual caruncle with opening of submandibular duct, then sublingual gland, submandibular duct, sublingual artery and vein along with submandibular gland, then facial artery and vein, then retromandibular vein, anterior and posterior branches, and then parotid gland. Parotid gland is something which is related to, a bit situated near the ear, and then parotid duct is also situated into that, and near that sublingual is there. So these are the various salivary glands. Next, salivary gland function. Different salivary glands in relation to the oral cavity. Bicuspid and molar teeth, grinding action. Incisor teeth, cutting action. Saliva entering mouth. And from the parotid gland is responsible for the secretion of salivary amylase. Submandibular gland, food particles progressively reduced and mixed with saliva. Tongue aids in mixing process. Then sublingual gland, below the tongue. So parotid gland is there, aromatic substances released from food. Then submandibular gland, sublingual gland, and these are the various salivary glands. Next, nerves regulation of salivary secretion. Parasympathetic nervous pathway for regulating salivation and demonstrates that the salivary glands are controlled mainly by parasympathetic nerve signals all the way from the superior and inferior salivary nuclei in the brainstem. The salivatory nuclei are located approximately at the junction of medulla and pons and are excited by both taste and tactile stimuli from the tongue and other areas of the mouth and pharynx. Many taste stimuli, especially the sour taste caused by acids, elicit conspicuous activity of saliva, often 8 to 20 times the basal rate of secretion. Also, certain tactile stimuli, such as the presence of smooth objects in the mouth, like a pebble, cause marked salivation, whereas tough objects cause less salivation and occasionally even inhibit salivation. So, higher centers of CNS get activated. Tactile and taste stimuli are the reasons for that activation, and then superior and inferior salivatory nucleus get activated, and that's situated near the pons and medulla, then in the junction, at the junction of medulla and pons. And salivary glands are activated, secretion or inhibition will be happening, and there are some many major points like acids, sour taste increases salivation, and when presence of smooth objects in the mouth is happening, there will be an increase in salivation. Then, which are the functions of saliva? Defense, antibacterial, antifungal, antiviral, immunological. Digestive functions, digestive enzymes, ptyalin and lipases. Ptyalin is something which is related with the digestion of starch, and lipases are the formation of bolus. Taste, protective functions, protective coating for soft tissues, protective coating for hard tissues like teeth. Lubrication functions, keeps the oral cavity moist, facilitates speech, helps in mastication and swallowing, and then there is one important function called the buffering function. When we just try to learn the formation and secretion of saliva by submandibular salivary gland, there is a region called striated and excretory ducts. Then end pieces will be there. Primary secretion is happening, then modification of ionic content is happening later. Contains amylase. In case of primary secretion, it contains amylase, Na plus K plus Cl minus HCO3 minus, and similar to plasma, but then the concentration occurs, like modification of ionic content is happening. Na plus K plus Cl minus HCO3 minus, etc. and added. So that's how saliva is produced. Salivation can also be stimulated or inhibited by nerve signals arriving in the salivatory nuclei from higher centers of the central nerve system. For instance, when a person smells or eats favorite foods, salivation is greater than when food that is disliked is smelled or eaten. The appetitive area of the brain, which particularly regulates these effects, is located in a proximity to parasympathetic centers of the anterior hypothalamus, and its functions to a great extent is in response to signals from the taste and smell areas of the cerebral cortex or amygdala. Salivation also occurs in response to reflexes originating in the stomach and upper small intestines, particularly when irritating foods are swallowed or when a person is nauseated because of some gastrointestinal abnormality. The saliva, when swallowed, helps to remove the irritating factor in the gastrointestinal tract by diluting the neutralizing the irritant substance. Sympathetic stimulation can also increase salivation a slight amount, which much less so than parasympathetic stimulation. The sympathetic nerves originate from the superior cervical ganglia and travel along the surfaces of the blood vessel walls to the salivary glands. A secondary factor that also affects salivary secretion is the blood supply to the glands, because secretion always requires adequate nutrients from the blood. The parasympathetic nerve signals that include copious salivation also moderately dilate the blood vessels. In addition, salivation directly dilates the blood vessels, thus providing increased salivary gland nutrition as needed by the secreting cells. Part of this additional vasodilator effect is caused by kallikreinin secreted by the activated salivary cells, which in turn acts as an enzyme to split one of the blood proteins in alpha 2 globulin, a form of bradykinin, a strong vasodilator. So parasympathetic nerve regulation of salivary secretion contains superior and inferior salivatory nuclei, tactile solitarius and facial nerve, submandibular ganglion, submandibular gland, chorda tympani, sublingual gland, parotid gland, otic ganglion, then glossopharyngeal nerve, tongue and taste stimuli. These are associated with that.